This screencast is on translation, where we go from the language of nucleotides into the language of amino acids to make proteins. In this screencast, we'll describe the 3 steps of translation, explain how a codon links the DNA code to amino acids, and explain how an amino acid chain is made in a ribosome. We've gone over this before, but one more time won't hurt. Transcription is simply transcribing a message from one form to another, like you would transcribe journals that are handwritten into digital form by either typing them in or scanning them in with and saving them as a PDF. You're just simply transcribing the information from one form to another, but the message stays the same. In transcription, we move the information, the message in DNA, into RNA, another form, but the message stays the same, and it's the same language, the language of nucleotides. Translation is when we move from one language to another. So we move from the language of nucleotides into the language of amino acids. Those amino acids will combine to make proteins. The genetic code is a rule. These 3 nucleotides, actually any combination of 3 nucleotides, will code for one specific amino acid. You can think of your nucleotides as your French to English dictionary. Now, in this French to English dictionary, we have codons, and these 3 nucleotide sequences are called codons. Here's our mRNA strand. The first 3 nucleotides, A, U, and G, code for a specific amino acid, and we call it a codon. So this is codon 1. The next 3 letters, A, C, G, are codon 2. The third codon is G, A, G, and so on, until the mRNA comes to an end. It's important to note here that there are 20 amino acids. Well, if you're good at math, you realize pretty quick there are more than 20 different combinations of nucleotides. So, looking at our codon chart, you can see that more than one codon codes for the same amino acid. These 3-letter designations stand for amino acids. I am not going to make you know the names of the amino acids. If you're interested, please look them up. How you read this chart. If you know the first letter of your amino acid, or the first letter of your codon, it's going to be in one of these rows. So, if the first letter is C, then the codon is going to be in this row. If the second letter is A, we go up to the top, and we can read the second letter, and this column is A. And then we pick out the third letter, and we have the corresponding amino acid. So this is our translation, or our translation key. This is our actual French to English dictionary. So let's talk about the steps of translation. Initiation is the first part, and this is where the mRNA strand, shown here in pink and purple, is going to attach to the ribosome. We have the small subunit and the large subunit. Now, this gets kind of complicated, and there's a lot more involved, so I'm keeping it simple. The next step of translation is elongation. We're going to elongate that amino acid chain. So write this down. tRNA brings amino acids to the ribosome, and they are attached in the order matching the mRNA strand. So here's a pretty overwhelming picture. Let's start at the top. The blue line here is our mRNA. Each of the little spikes here, the little teeth on this line, are nucleotides, and it shows the letter of those nucleotides here. our peach colored thing here, blob, is our ribosome, the small subunit and the large subunit. Excuse me. So here we have our tRNA molecule. The tRNA is made up of this light blue squiggly, an amino acid attached at the top, and a corresponding codon. So in our codon chart that we just looked at, UGC would code for serine. Now, the transfer RNA brings these molecules to the ribosome. If they match G with C, U with A, and U with A, this transfer molecule will attach, the ribosome will move over to encompass it, and it will create a peptide bond between the amino acids. The tRNA molecule that has just lost its amino acid is released. It will move back into the cytoplasm and attract another amino acid to be used in a ribosome in the future. Moving over here. Here's another tRNA molecule with an amino acid attached, and we see that the codons, well, they're complementary. So this tRNA will settle down on the mRNA, the ribosome will move over it, and create a peptide bond between these two. This process continues until the ribosome reaches a stop codon. And that's termination. There are 3 letter combinations, 3 codons, that actually code for stop. And by stop, we don't mean a special amino acid. We mean simply that the protein is finished. The ribosome falls off, and the amino acid chain is then free to be folded either by the ER or the Golgi apparatus. There are a couple of videos. I would like for you to watch these. They will help give you a visual of what's going on with this process and make it a little bit easier to understand.